Hello, my name is Sarah Novel, and today I will be presenting my seminar entitled Counterfeit Medicine Detection Technologies and Global Impact. In this presentation, we will explore how drug counterfeiting has transitioned from simple fakes into a highly sophisticated bioforensic threat and evaluate the modern scientific tools required to protect patients' lives. I will begin with an overview of counterfeit medicines and their global impact, then discuss the main detection technologies used to identify them. After that, I will highlight key challenges and the future directions before concluding the presentation. Counterfeit medicine have become one of the most serious challenges facing modern healthcare system. These products may contain the wrong ingredient, incorrect doses, harmful contaminants, or sometimes no active ingredient at all. As globalization, international trade, and online pharmaceutical markets continue to expand, counterfeit medicine have become a global public health threat affecting both developed and developing countries. The aim of this presentation is to discuss the global impact of counterfeit medicines, explore modern detection technologies, and examine future strategies for combating pharmaceutical counterfeiting. First, let us understand why this issue is important. Counterfeit medicine creates significant clinical risks. They may cause treatment failure, toxicity, and contribute to antimicrobial resistance when patients receive subtherapeutic doses of antibiotic. In addition, counterfeiters are becoming increasingly sophisticated. Modern counterfeit products often closely resemble authentic medicines, making detection more difficult and creating a technological challenges for healthcare professionals and regulators. Finally, counterfeit medicine generates major economic and social consequences by increasing healthcare costs and reducing public trust in healthcare system. Moving to the global impact, the burden of counterfeit medicine is especially severe in low and middle income countries, where regulatory systems may be weaker and informal pharmaceutical markets are common. The consequences include increased hospitalization, additional healthcare expenditures, treatment failure, and what researchers describe as poverty trap, where families repeatedly spend money on ineffective medicines and additional treatments. Counterfeit medicine also damage public confidence in healthcare system and legitimate pharmaceutical manufacturers. One particularly important consequence is antimicrobial resistance, or AMR. When counterfeit antibiotics contain insufficient amount of active ingredient, bacteria are exposed to subtherapeutic concentration. This promotes the development of resistant strains and contributes to the global spread of antimicrobial resistance. Counterfeit medicine may also mislead clinicians by making treatment failure appears to be caused by disease progression or drug resistance rather than poor medicine quality. Recent reports from France highlighted counterfeit oxycodone tablets contaminated with highly potent synthetic opioids known as nitazenes. These substances are significantly more potent than fentanyl and have been associated with fatal overdoses. Another major contributor to counterfeit medicine distribution is the rise of illicit online pharmacies. Research shows that many online pharmacies operate illegally or without proper regulation. During the COVID-19 pandemic, counterfeit drug markets expanded significantly as more patients turned to online purchasing. Criminal networks now use social media platforms encrypted payment system, and international shipping networks to distribute falsified medicine across borders, making regulatory enforcement increasingly difficult. International travelers are also considered a high-risk population. Travelers often purchase medications in unfamiliar environments and may not be familiar with local pharmaceutical regulations. High-risk medication categories include antimalarial drugs, antibiotics, and lifestyle medications such as weight loss products and erectile dysfunction treatment. The consequences can include treatment failure, exposure to toxic substances, increased medical expenses, and long-term distrust of healthcare system. To address these challenges, several detection technologies have been developed, such as field-based screening, laboratory analysis, digital traceability. The first category is field-based screening. Visual inspection remains the simplest method and involves checking packaging, quality, serial number, expiry dates, and product appearance. However, visual inspection alone is often insufficient because modern counterfeiters can produce highly convincing packaging. Portable spectroscopic technologies such as Raman spectroscopy and near-infrared spectroscopy provide rapid and non-destructive authentication by analyzing the chemical fingerprint of medicines. Mobile applications have also emerged as low-cost tools that allow barcode verification and smartphone-based detection. When field screening is inconclusive, advanced laboratory analysis becomes necessary. One of the most powerful technologies is liquid chromatography mass spectrometry. Liquid chromatography mass spectrometry provides highly sensitive chemical profiling and can identify active ingredient, contaminants, and impurities. A recent investigation involves counterfeit thyroid medications demonstrate that this technique detected major dosage inconsistencies and multiple undeclared substances, including steroids and other pharmaceutical compounds. Another important technique is gas chromatography mass spectrometry and is widely used in toxicology investigation and can identify hidden pharmaceutical ingredients within counterfeit products. For example, gas chromatography mass spectrometry was successfully used to detect undeclared venlafaxine and clomethazole in counterfeit herbal products marketed for alcohol withdrawal syndrome. In addition to laboratory method, new anti-counterfeiting techniques are emerging. Fluorescent tagging uses invisible labels that become visible under ultraviolet light, making products more difficult to counterfeit. Digital traceability systems such as a blockchain and Internet of Things allow real-time monitoring of pharmaceutical supply chains and improve product tracking. However, these systems have an important limitation. They can verify supply chain history but cannot independently confirm the chemical authenticity of the medicine itself. This leads to what I describe as the technological paradox. Advanced technologies such as liquid chromatography mass spectrometry provide exceptional accuracy but are expensive and require specialized laboratories. Blockchain offers strong security and traceability but cannot verify chemical composition. Raman spectrometry is portable and convenient but may have lower sensitivity than laboratory method. Similarly, mobile applications are accessible and low cost, but depend heavily on database quality and barcode accuracy. Therefore, no single technique can solve the counterfeit medicine problem independently. Looking toward the future, several priorities have been identified. These include the development of portable and affordable detection device, stronger international regulations, improved global collaboration, smartphone integrated spectroscopic tools, and enhanced pharmacist training. Pharmacists are uniquely positioned to act as supply chain and play a critical role in counterfeit medicine detection and prevention. In conclusion, counterfeit medicines represent a major global health threat. Modern counterfeit products are becoming increasingly sophisticated, requiring advanced analytical and technological approaches for detection. While laboratory technique, digital traceability system, and emerging anti-counterfeiting technologies have significantly improved detection capabilities, challenges related to accessibility, cost, and global implementation remain. Addressing this issue will require multidisciplinary collaboration among healthcare professionals, regulators, researchers, and policy makers worldwide. Thank you for your attention, and I would be happy to answer any question.